Hi there! You probably knew that our guts are inhabited by microbes like bacteria, archaea, viruses, and fungi. But did you know that they can influence your brain? Mind reading with Vera! Today's reading is about a study that investigated how gut-derived metabolite influences the brain of mice. Let's start from the beginning. Bacteria in our guts transforms parts of our food into different molecules, and we call these metabolites. An example of a metabolite is 4-EPS, which is a molecule that has piqued the interest of many scientists recently. There are some things we already know about 4-EPS. A bacteria in our gut creates this molecule from tyrosine, which can be found in certain foods, like fish and soy. The 4-EPS in our guts can enter the bloodstream, and that way it can travel into every organ in the body, even to our brains. Brittany Needham and colleagues decided to investigate 4-EPS. By genetic engineering, they have created two types of the same bacteria, one that would transfer tyrosine into 4-EPS, and one that was the same in all manners except it did not transfer tyrosine into 4-EPS. The scientists put these two bacteria in two different groups of mice that otherwise had no other bacteria in their guts. One group therefore had 4-EPS in their system, while the other did not. When the scientists looked at the brain activity of the two groups of mice, they have found that the mice that had 4-EPS had certain brain regions that were more active than normal. These brain regions were all part of the limbic system, which is the area of the brain that is responsible for fear and anxiety. The researchers decided to look deeper. When the scientists investigated the cells of the mice, they have found something interesting. Neurons are the cells in our brains that transmit information. The information moves faster if the neurons are insulated. Cells called oligodendrocytes are cells in our brains that are responsible for insulating the neurons. The scientists of our study have found that 4-EPS somehow stopped these oligodendrocytes from growing up, especially in the brain regions involved in emotions. Only mature oligodendrocytes can insulate neurons, and therefore these mice had less insulated neurons, disrupting the area of the brain that is involved in emotions. This led to behavioral changes in the mice. Mice that had 4-EPS were showing anxious behavior. Anxious mice like to stay safe. They prefer protected areas with shadows that hide them, while non-anxious mice like to explore their surroundings. When the two groups of mice were put into a maze, the mice that had 4-EPS spent more time lurking in the shadows, while the mice that had no 4-EPS decided to explore the maze. Therefore, the conclusion of the paper is as follows. 4-EPS, a gut-derived microbial metabolite, causes anxiety by affecting brain areas connected to fear. This way, microbes do in fact influence the brain. But don't forget, we are not mice, so further research is needed before we can draw conclusions in humans. I hope you had fun and learned something. See you in the next episode of Mind Reading with Vera!